Hi everyone, welcome to this M2D talk. Today we'll be talking about a causally inspired GNN for phenotypic driven lead design. Our speaker today is Guadalupe Gonzalez. She's a senior ML scientist in the frontier research team at Patient Design. Her expertise lies at the intersection of graph deep learning, causal inference, and drug discovery. Currently, her work focuses on causal graph deep learning for drug discovery, going from small scale to larger scales systems. And she had her PhD from the Imperial College, advised by Michael Bronstein and Kirill Visikov. Thank you so much, Guadalupe, for accepting our invite and presenting your work with here today, and looking forward to it.
Yeah, thank you for the introduction and thanks for inviting me. So thanks everyone for being here. Today I'll be talking about PDGrapher or model, causally-inspired model for phenotype-driven lead design. And feel free to ask questions as we go. I think I prefer very much that kind of format. So just raise your hand or write, and I think I should be able to see the chat or raise hands as we go. Okay, so I'll go ahead. First, I wanted to give a brief introduction or a couple slides into the difference between phenotype-driven versus target-driven drug discovery to see what the context is that our project is. So there's two major approaches to lead discovery. Target-driven drug discovery is based on like having a specific drug target. Usually this is a protein that we know is involved in disease, and we're trying to develop a specific small or large molecule that would perhaps bind to it and exert a specific effect. And so the key element here to assess which one of these molecules or therapeutics are potentially good are target-based assays, where we directly measure either the binding capability or let's say, even the potency of that binding. And when we have a good result here, then we said that we have a lead, which means a molecule, small, large molecule, an agent that can be potentially optimized to then go into preclinical and clinical trials. So that's what a lead is. It's a agent that shows some promise to be a potential therapeutic. The second largest approach to lead discovery or drug discovery is phenotype-driven discovery, where the key element is a disease model. So this is a model that represents either the whole aspect of the disease or certain aspects of the disease. And this can be animal models, cell models, and even human models. And so here, whenever we have certain candidates that we think might be effective, we have phenotypic assays. So these measure what happens in this disease model when we administer each one of these agents. So ideally, we would see that the disease model transitions into what would be a healthy state, or we have a response that we're looking for. So whenever, again, here, through scanning libraries of compounds, we get potentially one that looks promising that we call a lead. And typically we would go through a target identification phase, just because identifying the targets help us know about the side effects and off-target effects, so that can give more information about the specific compound and whether it might fail due to toxicity reasons later on. And again, then from here on, we will go to the same process. So since the beginning of drug discovery, let's say, phenotypic-driven approaches were dominating, especially when we're thinking about like natural compounds. These were discovered through seeing what their effects were in specific systems. So there was no information about what their targets were, or no, like, in a way, targeted design. But since the advent of high throughput technologies, as you might be aware, target-based approaches became the most prevalent, where we have, specific molecule, and then we try to develop an agent for that. However, given or with the observation that between 1999 and 2008, most of the first-in-class drugs approved by the US FDA were discovered empirically without actually a drug target hypothesis. Phenotype-driven approaches started to regain importance and interest by the research community. So our work, PDGrapher, is situated here in the context of phenotype-driven lead discovery. So typically how the problem formulation works is we have a system. Here, when I talk about phenotypic state, I will be talking about gene expression. If you're not familiar, gene expression is, in layman terms, the level of the gene that we have in a specific cell or tissue or system. Typically, this is very localized, and what this means is that it would reflect the level of proteins that that gene would translate into in the system. And in computational terms, I guess this is a number, a real number per gene. So what we would have is, our list of genes and a real number representing the normalized level of these genes in our system. So when we have a healthy system and we take a sample, we can have a picture of the healthy signature, what we would call the healthy signature. And so this is what we see here in green. So this is what we would see here in green. And then we would have in our system a disease, which is represented as perturbations here. So this disease, let's say, that interferes with the function or even folding of the proteins that these genes encode. So that causes changes in the gene expression. So this is what we would call a disease signature or a disease phenotypic state. So the goal in phenotype-driven lead discovery is usually then to find a specific intervention or a specific perturbagen that would reverse those effects. So that's what we would call a treatment. So a treatment acts in a similar way as a disease. At the end of the day, they are perturbations or interventions in a system. And again, we have a modified or a different gene expression here. So the goal is for the blue gene expression to be as close as possible to the green gene expression. So we can look at how existing work tries to tackle this. We have here, of course, our key element or phenotypic assay dataset, where we have replicates of our disease model. We have a library of perturbagens, meaning when I talk about perturbagens here, that is any agent that is able to modify or affect or perturb a system. So this is, in this case specifically, we're talking about chemical compounds or genetic interventions in these systems. And we have a set of treated samples that these perturbations generate. So ideally, what we would be able to do is take our disease model, check experimentally what happens when we treat this disease model with our whole library of perturbagens individually in combinations of 2, 3, 4, and so on, and then see what would happen and then see which one gives us the desired response. However, in reality, this is pretty costly. So the community has resorted to modeling this using machine learning. So what we would have when we have a specific disease model is we predict what happens, so what the response is upon each in single one of these perturbagens. And these, the kind of models that do these predictions are probably known to many here. These are the response prediction models, where we have a system and try to predict what the response is to a specific perturbagen or compound. And once that we have the response for each of them, we check the similarity between each of these responses and the desired response. So this could be anticorrelated, a response that is anticorrelated with the disease. And once, let's say that we find a response that is most anticorrelated with disease, then we say, okay, the perturbagen that generated this response is my lead compound. And then I can move forward with that. So this is what we would call here indirect prediction of perturbagens. I have to go all over my library, check what the response is, and then from that select the perturbagen. So while we were discussing this problem, we wonder if it was possible to do this prediction directly instead of having to predict responses for all the perturbagens in a library. So the goal here would be to have a disease sample and a target treated sample that we want to, like whose state we want to achieve, and for a model to predict exactly what perturbagen is necessary here for this transition. And so this is what we call a direct prediction of perturbagens. And this is exactly the problem that PDGrapher tries to solve. So I'll go next into our problem. Yes, I see a hand raised. Prudencio.
Yes, do you mind going back to the previous slide? I was just wondering,
Sorry.
yeah, I was just
Mhm.
wondering if the goal is to predict the exact perturbagen or a distribution over a set of perturbagen that you have in the library, for example.
So if the goal is to predict a distribution over this or just a
Yeah,
specific one?
exactly.
Yeah, so the goal is to predict the specific one. Of course, this is a extremely challenging problem, and we will see in the results that there's a lot of room for improvement. Specifically in the way that we have formulated it, it's like a ranking of genes because we represent these perturbagens based on the genes they target. And so, there's a matter of, okay, where is the cutoff here? Like, how do we exactly define this? But how we formulate it is we want the specific perturbagen. So we don't want to rely on the libraries.
Okay, perfect. Thank you.
Okay, I'll go to the next slide. Okay, so next I'll go into the problem formulation. And because we rely on causal inference to formulate our problem, I have added like a brief intro to causal inference, which I hope will be of help in understanding the problem formulation. So specifically, we use structural causal models. So causal models are mathematical abstractions that quantitatively describe the causal relations between variables. And specifically, structural causal models, or SCMs, consist of a causal graph. So we see here in this system, we have 4 variables, and the edges or links represent the causal relationships between them. So in this case, it means that V2 causally depends on V1, and V4 causally depends on V2 and V3. I haven't plotted or drawn them here, but it is also assumed that each one of these variables is dependent on what we call an exogenous variable. So V1 would be depending on a variable called U superindex V1, V2 would depend on U superindex V2, and so on. And so this causal graph is paired with what we call structural causal equations, which are down here. And these are deterministic functions that encode or encode the value of each one of these variables as a function of its parents. So here we see that V1 is a function of U superindex V1. So we see there's no other node that is pointing towards V1. And then here, V2 is a function of U, V2, and V1, which is encoded in the graph. So SCMs are really useful because they allow us to represent interventions explicitly. And by interventions, I mean any kind of perturbation in the system that sets the value of these variables. And from what we were seeing in the previous slides, both, let's say, genetic knockout experiments and drug compounds administered to a system are interventions because they might bind to specific genes and explicitly change the value of the gene expression of those genes. So how we represent that in SEMs, we can represent those through the do operator. So when we see this do v2 equals lowercase v2, what that means is that we are assigning the value of v2 equals to an arbitrary value. So if it's a knockout experiment, this would be 0. And that is also represented in the graph through mutilations. So we see here that when we intervene on v2, v2 doesn't depend on its parents anymore. Whereas before it depended on v1 and its external or exogenous variable, now it doesn't depend on anything else but the intervention itself. Okay, and why it's important to explicitly model these interventions will be a bit clearer in this slide. So we can think of SCMs as generative models, where here I have instantiated very simple structural equations where v1 and v3 are just equal to its exogenous variables, and v2 and v4 are just a logical AND function. So when we will go recursively through this graph, this acts as a generative model. So we can generate samples following from these structural equations. And so we did that, or I did that for this table. And here, after doing this, we obtain what's called an observational joint distribution. So this is whatever observations or data we get upon no intervention. And then when we look into the system that has been perturbed, we can again generate samples following the modified structural equations. And here we obtain what's called the interventional joint distribution. So what we can see here is that these joint distributions are different because we have broken the relationship between V2 and its parents. So it's important for us to model interventions explicitly because let's say that we have a model that learns from observational data, or it doesn't have to be observational, it might be data from different interventions, let's say different drugs. Then when we try to predict for drugs that we haven't, or compounds, perturbagens that we haven't seen, then our models won't generalize very much because they are out of distribution. So this is the big problem, or one of the big problems when we're trying to learn from interventional data in biology, and especially in the case of this phenotype or these phenotypic assays. Okay, so that was the intro. So now going into the problem formulation, we assume that we have an SDMM where we have exogenous variables, V, which are the variables in our system. And in this case, we have genes, F, which are the structural equations, and P of V is a specific or certain probability distribution over E. And this is an SEM associated with causal graph G. And we have a data set with tuples where each tuple is a combination of a healthy gene expression, that's V super index H, and this is one value, one real value per gene. And we have in this healthy system a specific, disease that is perturbing my system. It's represented as a set of nodes that are being, affected by the disease. So, for example, if it's cancer, like specific type of cancer, this would be the cancer driver genes for that specific type. And here, and that leads to the disease node state, so V super index B. And here, G super index P for the tuple is the mutilated graph upon this intervention. So this is the disease mutilated graph. So our goal here is to find this intervention set, U prime. So this would be the set of genes. It can be one or it can be multiple genes that or a perturbagen has to target to achieve this treated gene expression state, P superindex C. And ideally, this treated state would be as close as possible to this initial healthy state. And again, we note here, G superindex C prime is the treatment mutilated graph. So it would be the graph where we have mutilated, in addition to the disease mutilations, the mutilations based on the treatment intervention. So we can represent this goal in this way. We're looking for the U prime that maximizes this probability over the disease mutilated graph of node states being equal to the treated state, meaning this final state, given that we intervene on U prime. So let's say I have this system that I know has this, mutilated edges because it's a disease system, and I'm trying to look for this U prime that leads me to the treated state. Now, these interventional probabilities in practice are extremely challenging to compute in the cases where we don't have a causal graph or where we have noisy, incomplete data sets, which are actually all of these conditions are present in this problem. So we make some assumptions, and the first one is that we don't have unobserved confounders. And this allows us to transform this interventional probability into a conditional probability over the mutilated graph upon the interventions. So here we forego the do operator on this U prime by computing these probabilities over the mutilated graph when we intervene on U prime. And so this is this is just thanks to the theory on causal inference that says that this conditional probability is equal to this interventional probability in the absence of unobserved confounders. So just to recap, what we have is the goal that we have is to find a perturbation set U prime with the highest likelihood of achieving the treated state, P superindex C. And so that's represented in this equation, which under the assumption of no unobserved confounders transforms into a conditional probability upon the in graph that has the mutilations due to these interventions. And so first, we formulate this problem in the causal learning realm, but then the second step that we take to be able to address this from, let's say, the practical point of view, or to address some of those concerns given by the lack of a causal graph and noisy and incomplete data, is that we jump to the graph representation learning or the representation learning world, and in which our goal is to find a function f that takes a graph, the mutilated graph that we have here, the disease states, the treated states, and it finds that U prime that maximizes the probability over the graph GP prime, given the disease state and the intervention itself. So here, the reason why we say we jump into the graph representation learning, or why we use GNNs, is because we have to compute probabilities over a graph. And what this means is that each node or each node's value depends only on the parents. And we know that GNNs compute transform representations based on the values of the parents. So this seemed like a very natural way of encoding this, or I guess translating this into graph representation. And I think, yes, there's a hand raised. Jason?
I just want to make sure I'm following. So you're saying we're going to try and do inference over the graph or over the over U?
We're trying to do inference over the graph. So this the bar represents a condition on U. So given that we see the value of U being a specific or U being a specific set of genes.
And then you're saying you're trying to find the graph such that the conditional is equal to the interventional distribution or something else?
We're trying to find the U prime. So we're trying to find that U
Yep.
that for which these probabilities computed over the graph are equal to the treated state.
But if we don't
So let me
know,
go.
if we
Mhm,
don't know the causal graph,
mhm.
I guess what is the and we're doing inference. So if we're doing inference over the graph and inference over U, like in what sense is there a causal connection at all, really? You know, it feels like we're not leveraging any structure about the distribution. Am I missing something?
Yeah, so I mentioned that we don't know the causal graph. So we
Yep.
assume that we have a noisy and incomplete version of the causal graph, in which in this
Okay.
case we use PPIs and gene regulatory networks as two possible noisy causal graphs, just because it's not even possible for us to do causal graph discovery in this case because we don't have ground truth at the large scale that we need.
Okay. So you using you're thinking of like your protein-protein interaction or the like as a prior over the graphs, and then you're saying okay, given that prior, I'm going to do inference over the graph and then also try and find U that maximizes the probability of observed variables that I've seen.
Yes, so
Or
I guess when I say inference over the graph, the only thing we're trying to find is that U prime. We assume that we have that prior over the graph, but we are not trying
I
to
see,
modify the
I see.
graph.
I see. Okay, so
So
we're
when
just
I
sampling
say
from that prior, or
we're just using it as the support for these probabilities. So what this means is that, like, what this super index means is that, let's say, kind of goes maybe back here. If I'm trying to compute the probability of V4 being
Mhm,
equal to its treated state, that means that the
mhm.
probability is only over V2 and V3. And,
Okay,
V4's exogenous variable.
there's only a single graph here. It's not like we have some distribution over graphs. We have a single graph, and we just want to do inference over U under that graph, but you're calling it a prior because you're kind of not sure that it's the correct graph.
Yes, exactly. And we do have some minor modifications between samples because we have these mutilations. So
Mhm.
there might be this minor, well, depending on the degree of the node that has been, perturbed, might be many nodes that are mutilated, but the graph is always the same except for those mutilations.
Okay. Okay. There was a question in the chat as well. Thanks.
Okay, thank you. Um, so Sohib says, I'm paraphrasing the insight here to make sure I understand it. Is the insight here that we can interpret interventions as changes in the causal graph? So we want to use trained models that are model agnostic in the sense that they take a graph as an input and not learning the graph from data. So we can give a graph to the model ourselves that represents a successful intervention. Or models that are graph agnostic. So primarily, the effect of the intervention is on the values of the nodes. And that's exactly what we're interested in. So the interventions will change the graphs, but these changes in the graphs are known, as in they almost come with the intervention itself, because they will be mutilations on the incoming nodes that are perturbed. So the main goal, or the main thing that we're interested in, is the states of the nodes, not the states of the graph. Does that answer your question?
Can I just ask a follow-up to that? You said the thing we are interested in is the state of the node, not the state of the graph. But when we do the intervention, we at least we get data about the state of all the other nodes in the graph, right? And that's what we use as data set for the training.
Can you repeat that?
You said we are not interested by the state of the graph, but the state of the node. And I was saying that when we do the intervention, the whole graph change. At least the set of all the other nodes graph change. And we measure that, and that's what we are using for the training and for the inference of the minimal set U or U prime. Am I correct?
Yeah. Yes. 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 Yeah.
Zofia, please.
So those changes in the graph will also be like partially affecting the treated state, so the state after the intervention. Yeah.
If I could clarify what I was saying earlier, I think, and do correct me if I've misunderstood or understood something separate here, but I think like the values of each node represent like gene expression or something similar that we could experimentally get. And the graph is something that we give to the model. So we would write that down and say like, hey, this is what we think is happening. Take it. And because of causal inference, you know how an intervention is going to change it. So what you're doing is you're searching over these graphs by giving it to the model and seeing if the model can. Well, okay, that part I haven't fully understood, I guess, but it seems to me like that the insight, at least so far, was that we want to use a model that's predicting the values of gene expression, for example, without like accidentally learning some graph structure implicitly, because we want to explicitly give it a graph structure that represents an intervention. And I think that's what I'm understanding so far.
Yeah, I think there maybe was a disconnect in how I understood your written message. But yes, your understanding is correct. So we're trying for the model to learn the structure of the graph, but actually how we are representing it is through this set of interventions, because they are equivalent. So if I say that U prime is equal to V4, what that means is the graph will be the initial graph minus these edges that are incoming on V4. So you could interpret it as, yes, we're trying to learn a graph structure. In practice, what I'm doing is I'm trying to learn the set of variables, but they are equivalent. So yeah, they are the same.
Okay. Okay, I feel like at least I understand enough to continue paying attention to the talk. I'm very this is tough stuff for me, I think.
Okay, there's another comment in the chat. In the objective function, do we know the ground truth, or is it the same as VH we have in the data tuple? That's a good question. I mean, all questions are good. So here, yes, we know VT. So there's two possible scenarios. One is where VT is exactly VH. And in that case, we're trying to learn exactly how to go back to the healthy state. In our case, VT is a treated state, not necessarily VH, so that we could use or take advantage of all the like perturbation data that is available. Because let's say for the case that we wanted to learn explicitly how to, go back to VH, there's not a lot of data that we can learn from. Of course, there's all the replicates we can have of the healthy state, all the replicates we can have of the disease state. But if we're dealing with one disease, for the model, it's hard to learn just from a small data set. So how we use the data to learn here is we have these all the perturbation data sets. So these libraries of perturbagens and their responses. And then we use that as the treated state. So short answer, the treated state is always known, and it could be either post, chemical or genetic intervention, or it could be the healthy state. Okay, so I'll move ahead. And again, keep asking questions if there's something that's not that can be clarified. Okay, so I believe I was at this point. So we're trying to move from the causal inference formulation to the representation learning field. So just to clarify, maybe one last time, that this probability over the graph represents the factorization of the probability over the parents in the graph. So graph representation learning, specifically GNNs, are very suitable for this because they compute values of the nodes or probabilities, meaning the final labels based on the values of the parent. So given this goal, which is the U prime that maximizes this probability, intuitively, we would need to search the space of all interventions, U prime, so that U prime here is the unknown, and then score how good they are, meaning this probability. So let's say we have all the possible genes and we have we predict this probability given that U prime is equal to gene 1 or equal to gene 2 or gene 3 and so on, and try all the combinations, and we can have all the values of this probability. So then finding the U prime that maximizes this probability is very easy, right? And this is exactly how the response prediction models work, where they're trying to just go through the library and then predict the one that has the best score. However, as we have seen before, this becomes quite intractable. So what we propose is a smart way to search the space of intervention, the space of U primes, through an intervention discovery module, and then a way to score each intervention. So a way to compute this probability P over the mutilated graph. And I think I saw Jason.
Yeah, is it on graph neural networks? Isn't it only the case they only depend on the parents if you use a single layer GNN? Like if you use a multiple layers of GNNs, you aggregate from further hop neighbors. Is that the case, or what am I missing?
Yes, yes, that's the case exactly. Yeah.
So you just use a one layer GNN. Is that the idea?
Not exactly. So we did a hyperparameter optimization in the end, because we have to remember that we don't have the perfect causal graph. So ideally, if we have the perfect causal graph, one layer GNN should be perfect. We, just get the information from the parents, and that's just enough to compute the values of the children. In this case, because we don't have that, we tried with 1, 2, or 3. I think that was the most that we tried.
Did you try like literally just training kind of separate architectures for these different models, right? So you could imagine each mechanism in the causal graph is represented by a small neural network, and training in that fashion. Because the tricky thing about GNNs is that like they kind of mix together information sharing and capacity. And so you could have like you may be needing to do hyperparameter tuning because you don't have enough representational capacity, and not that the information sharing is the issue. I don't know if you had to look into that at all.
Yes, that is a great question, because there was this paper on the neural causal connection that tries to, fuse neural networks and SEM, so this type of models, and they have one neural network per node. So each node has its own neural network that operates in the parent. So in that way, you have like the perfect capacity, right? Because each node has its own structural equation, which is what the SEM represents. Each node has its own function. In this case, we wanted to keep that model. Of course, it's not scalable at all. We have, I think, over 10K nodes. So, it's not tractable to think, okay, we might have, over 10K, let's say MLPs, even, one layer neural networks. So we want to keep the tractability of GNNs. But what we do to increase the capacity and try to have different structural equations for each node is we add learnable embeddings. So let's say we have gene A, and our gene expression values go from 0 to 1. We bin them into n, not equispace, but based on the like the percentiles of the data, and we assign a learnable embedding to each of them. And we share this across all of the samples. So for sample 1, 2, 3, 4, 5, n, they will have the same, gene A will have the same learnable embedding if it has the same value. And then we have different learnable embeddings for each one of for all the other genes. So this adds a lot of representational capacity. And in fact, without this, it wasn't really working very well.
Gotcha. Okay.
Yeah.
I want to do a quick time check to make sure that we maybe pause on the question a bit and allow you to progress and take some question more at the end.
Yes, that's a great idea.
Cool,
Thanks.
thank you.
Okay, go next. Okay, so I think what the following slides are, I think, way less dense, so I should move through them faster. So this is how our model looks like. We have our disease gene expression sample here. You can see this is, one value per gene, and we overlay them over this proxy causal graph. We have the specific perturbagen, which shifts the gene expression from disease to treated. And again, this is represented the same way. So what PDGrapher does is it takes the initial disease sample and the final treated sample, and it makes this combinatorial prediction of perturbagens. So it tries to search for the perturbagens in a smart way. How this is optimized is we start here from the diseased sample and the treated sample, and this perturbation discovery module proposes a candidate U prime, and then we go again from the disease sample and this candidate perturbagen, and the response prediction module tries to predict the treated response, which should be as close as possible to the real treated response. And how, again, we represent mutilations in the message passing is just by removing these edges from the GNN message passing. So that's pretty simple in a way. Okay, just to recap, this is how existing work looks like, where it tries to check predictions for all the compounds in the library, and PDGrapher tries to directly predict this perturbagen that would shift gene expression from an initial state to a treated state. Next, I'll go into datasets, experiments and results. We have single gene knockouts and compounds from the L1000 dataset. And to build the proxy causal graph, we have a collection of BioGrid, this dataset, and the human reference protein interactome. So we have around 10,000 nodes, 150K edges. So you could say a pretty large graph. We have two training settings, one where we pulled out novel samples. So these are replicates that can be replicates of the same perturbagen or completely new perturbagens. And we also have a setting where we train in one cell line and predict for a different cell line. So if we train on lung cancer, we predict for breast cancer and vice versa. We have 4 baselines in our initial work, 3 mechanistic baselines, one, okay, so maybe before explaining the baselines. So what our model predicts is, I mentioned this before, but I'll mention it before the baseline. So PDGrapher at the end predicts a ranked list of genes where the top N predicted genes are those that are selected as the members of U prime. So are selected as the perturbation set. So the random baseline ranks these genes randomly and then selects the top K. The cancer targets baseline at the first N genes as the target of existing cancer drugs, and the remaining ones are sorted randomly. The cancer genes does a similar work, except for the top ranked genes are the ones that are, cancer associated, so cancer causing genes. And finally, we also compare with Estigen, which is a response prediction baseline. And we are working now on potentially adding more response prediction baselines, but this was a good baseline because we could use it with both chemical and genetic data sets, and it works in known samples and or known cell lines and new cell lines. So it worked on all the settings, so it was a good baseline to include. And I'll discuss the results next. So to understand the metrics, here we have how the result would look like, or the output of these models would look like. We have our ranked list of genes, and what we want is for the ground truth targets to be at the top of this list. So if the ground truth target is predicted at the top, we say that that's a ranking of 1. And if it's predicted at the bottom, that's a ranking of 0. And here we show the results for both the lung dataset and the breast dataset. And these results throughout the presentation are on chemical datasets on the random splitting. So the one where we have new and seen perturbations, just for like simplicity. You can look at or I'll mention briefly how results compare with the other settings, but it's relatively similar. So we have here for chemical, lung and breast datasets, where we see that PDGrapher ranks the ground truth or the perturbation, the correct perturbations at the top of the list. And again, we have a second metric for this ranking where, let's say that we have 2 targets that are in this perturbation set U prime. And let's say that they fall in position 1 and position 4. So because we have 2 ground truth targets, we look at the top 2 predicted genes. And if at least one of them is in the correct set, is a ground truth target, we say that that's a partially accurate prediction. So we use this to compare the predictions across models. Okay, so we can compare these across the mechanistic baselines and Estigen. And again, here we see that PDGrapher outperforms the baselines. However, here is where we can see that these like accuracy numbers can be improved like massively. It's promising in the sense that we show that it provides a set of set of genes that are maybe more informative than baselines, but still there's a long way to go here. Interestingly, we also show that the predicted genes by PDGrapher are closer to the ground truth targets or closer than what would be expected by chance. So the x-axis here is the shortest path length between the predicted targets and the ground truth targets in the network. And this is interesting because we know that genes that are closer in the network tend to be more similar functionally. So this is like an indication that the predictions are functionally similar, even if they might not be exactly the right or specifically the correct gene. And another benefit of doing this direct prediction as opposed to the indirect prediction of perturbagens is that the training is much faster, specifically comparing with Etigen. In the chemical data sets, our model train 15 times faster than Etigen. We also wanted to see for specific predictions, how for a specific example, how these predictions look like. So we looked into raloxifene, and here we have plotted the spatial enrichment around the targets, the ground truth targets here on the left, and the predicted targets. And here we see that estrogen receptors 1 and 2 were predicted correctly, but the other two targets were not predicted by PDGrapher, but instead it predicted SHBG and PDE5A. However, we see that the spatial enrichment looks quite similar, except for this enrichment around PDE5A. When we look a little bit closer into the predictions for raloxifene, which is a second generation selective estrogen receptor, here we show in squares the targets that were correctly predicted, so the estrogen receptors 1 and 2. The triangles are the targets that PDGrapher didn't pick up, and the circles are the targets that it predicted instead of the triangles. And so a way that could, or specific mechanism that could explain these predictions is that raloxifene has been documented to increase the levels of SHBG in 3 different cohorts. So that could explain the relationship between raloxifene treatment and the modification of SHBG's expression. And additionally, for PDE5A, its prediction could be related to the estrogen or to its relationship with estrogen receptors, because we know that estrogen receptors increase vasodilation, whereas PDE5A decreases vasodilation. And so potentially raloxifene treatment, which modifies estrogen receptors, could have an effect on PDE5A. So the takeaway from our work is that we tried to reformulate or look into a fresh perspective into phenotype-driven discovery. And we tried to reformulate the problem and propose PDGrapher for the prediction of therapeutic targets, which outperforms mechanistic and traditional baselines. It performs direct prediction of perturbations, is computationally efficient, and it illuminates the mode of action of predicted perturbagens. And maybe this is the last slide with some takeaways. If you're considering using PDGrapher in your research, there's a couple of things to note. There's 3 ways that I think this might be used in your research. So if your dataset distributions match what PDGrapher was trained on, you could reuse the trained model. If the datasets come from a different distribution, PDGrapher could be retrained given specific considerations. And finally, if you don't want to use PDGrapher, you don't like the model, I think there's also value in the problem formulation, in the sense that we're trying to tackle the problem from an inverse perspective, so trying to do the direct prediction versus the scanning of the library. So there's also room for other models to be proposed here to actually address that problem formulation. And here I end.
Perfect. Thank you so much for the talk, Guadalupe. There's time for more questions. We can maybe start with those in the chat, and if you have any question, please raise your hand.
So let me go to the chat.
Tristan, I think really that, but like, I think the last question was from, I think it was Richie actually.
Which one?
Richie, I guess. Richie Gupta.
Uh-huh. I think I answered that one. The one about the objective function. Is that the one? Yeah. Okay, so if Tristan is not here, then I can go to... Yes. Go ahead. Where does the initial causal graph for the healthy or disease states come from? The initial causal graph for the healthy comes from these libraries of protein-protein interactions, or we also use gene regulatory networks from the healthy samples. So these are computed using a specific causal discovery algorithm from the gene expression samples themselves. So we have the initial graphs for the healthy cell lines, and these are then mutilated based on the disease state. So we find in Cosmic the disease driver genes, and we use those to mutilate the graphs. So those would be the graphs for the disease cell lines. Okay. Jepeng. Hi, Jepeng. Nice to see you here. Could you comment on the size of combinatorial perturbations and how that might affect model performance? Do you mean to ask the size of the U prime that we need to predict?
Right. Hi, Guada. Yeah, I mean the size of, for example, how many genes you're perturbing, the kind of correlations with how the model performance is like.
Or like if in our training set we have perturbations that have massive sets of genes that they are targeting.
Yeah, for example.
Yeah, that's a really great question. And I think most of our, of course, the genetic data sets are all one gene because they are single gene knockouts. So we don't see, any difference there. In the chemical data set, we also didn't have massive sets. Maybe the largest one was 50 or 100, but still they were all within, the smaller range. I think the more genes we have, I think the harder it is for the model to get them all right. But the fewer we have, let's say if we have just one, it's also extremely challenging for the model. I think for two reasons. One, if we have a single gene target, the effect on the system is not that large, actually. We saw from the genetic data sets that the effect after the knockouts was smaller overall or on average compared to the chemical data sets. And this has also been documented before. So I think a middle ground is probably ideal, but I haven't looked into model performance as a function of the perturbation set, which is a really interesting thing to look at. Let's see. Rochelle, for this work, the interventions are hard interventions. Yes, they are. So gene knockouts set the gene expression to 0, and chemical interventions set the gene expression to the post-intervention value. So this might not be 0, but it's a specific value. So yes, they are hard interventions. And then question 2, if I understood correctly, with the method, our search space is confined to the observed perturbations in the library, or can the method propose novel combinations? So the targets that it proposes are completely arbitrary. So the perturbation discovery module scores the gene. So it learns this during training. So it doesn't rely at all on this, existing library. It can predict any combination or any single target. And that was, like one of the benefits or one of the reasons why we wanted to propose this, so that it doesn't have to rely on the library of compounds.
I think the question by Tristan wasn't answered, if you don't mind.
Oh, I thought you had mentioned he had left the call.
I can ask my question. I'm still
Oh,
here.
sorry.
No problem. So my question was, when you're doing this maximization over the interventions, so you're talking about you have access to the graph, but do you only have access to the graph structure, like these protein-protein interaction network? Do you only have a way to know which protein are interacting together, or do you know how they are interacting together? And the question was, like, because you need that probably to evaluate the likelihood that you're maximizing over.
Yeah, that's a great question. For the protein-protein interaction network, we only took the information of the structure. So we know they are interacting. We don't know if it's positive, negative, and in most cases, we don't actually have directionality either, which is one of the core things we need to have this recursive system. And so the idea here was to use the GNN with the added the representation power from the learnable embeddings to learn those mechanisms from the data without adding that prior information, because I didn't look exactly into that, but I'm sure that that's not known, or that would be, I have a pretty certain guess that wouldn't be known for all of the interactions. But yeah, that's a great question.
That makes sense. Thank you.
Yeah, thanks for your question.
One last
Yipeng?
from Yipeng before we close.
Sorry, yeah, super random question. It's more of an imaginative one, because all of your data sets are on cell line levels. And I'm wondering in real world patients, for example, when you don't really have what the healthy patient's signature should be, how do you know at inference time what is the correct input for the model? I was thinking perhaps considering a distribution of healthy patients, well, healthy people would, well, or to rephrase, making the model possible to accept a distribution of healthy patients' signatures as input might be a better approach, or if the model can be adapted in such a way.
Yeah, that's a fascinating question, and we have discussed that internally because we have some patient data, and some of the things we have discussed is potentially, like you say, have some representative healthy states. Other things we have discussed is having, for example, samples from, because it depends on the disease. So for example, if it's a tumor, you could have samples from the same tissue, but non-tumor. So that could be your healthy sample. Or, I mean, it might not be ideal, but from a similar tissue or area. So there are different ways that you could build these healthy goals that could be would facilitate using this with patients. So I think that what you mentioned about the distributions of healthy patients sounds like a really good idea too.